So continuing with translation, so these are the steps which we discussed earlier in the earlier video. So methionine is the first which is incorporated in every protein. In the case of bacteria, it is formyl methionine. So these are the events which happen. So as we said earlier, you can also see the video which is being played here. That is IF3 displacement causes the 50S to join the 30S. As a result, the GTP of IF2 is hydrolyzed and IF2 GTP is leaving the complex. So this is what is happening, and this is the end of the initiation of translation. Coming to the next one, which is elongation. So in elongation, so what should happen next is the next amino acid should come and a peptide bond definitely should be formed. So the first step in that elongation is elongation should happen. The next tRNA should come, and the next tRNA is welcomed by the next site towards the left, which is called as the A site, that is the amino acyl site. So here what happens is the first step is the amino acyl tRNA selection. So that is nothing but the entry of the second amino acyl tRNA to the A site, which is the first step of elongation. So here the amino acyl tRNA, so here also you have some factors which are which helps in elongation process. The factors or the proteins which help in initiation, we call them as initiation factors IF. Here you have the factors which help in elongation, and they are termed as EF. And the first of the elongation factor that comes to play in elongation is EF2 GTP. This EF2 GTP is attached to the tRNAs. To the it will not be get attached to the initiator tRNA. With every other tRNA, you can see this particular protein attached, which is called as EF2 GTP. So this EF2 GTP is attached to every other tRNAs, and every tRNA will have a chance to enter into the A site and check whether a complementary sequence is there inside the A site. Because as with the P site, there will be 3 codons, or there will be a single codon with 3 nucleotides which is present in the A site also. So every tRNA will have a chance to enter the T site and see whether they are having complementary bases. So every tRNA which is entering the A site will be having the particular elongation factor, which is called as the EF2 GTP, and this GTP will be hydrolyzed whenever correct codon anticodon pairing happens or complementary pairing occurs. So this particular EF2 will leave the particular tRNA only when the correct complex or correct pairing occurs, and when this pairing occurs, EF2 GDP will be released from the ribosome, and this EF2s are replaced, because for every tRNAs EF2 GTP is required, and this is being replenished with the help of another elongation factor which is called as EFT. So now we came across 2 elongation factors, EF2 as well as EFT. EF2 is combined with the tRNA, and EFT is an elongation factor which is not present inside the ribosome, or it does not associate with the ribosome. It is seen outside, or it is seen in the cytoplasm, and its function is to regenerate the EF2 GTP to EF2 GTP, GDP to GTP. So next is the peptide bond formation. That means after the 2 tRNAs are present at the A and P site, which has formed complementary pairing at the mRNAs. Now the peptide bond should be formed, and the peptide bond formation is catalyzed by the enzyme which is called as peptide transferase, which is nothing but an rRNA which is seen in the larger subunit, or which is seen in the 50S. That's why it is called as a ribosome or an RNA enzyme which catalyzes the peptide bond formation. The tRNA which is at the A site, after this process, it will be attached to a dipeptide. So that can be seen in the video here. So in this particular video, you can see how the peptide bonds are formed. So here you can see the second position and first positions are marked here. So what happens is one amino or one tRNA is there at this particular P site, and the other one comes at the A site. After that, a peptide bond is being formed. So this is the location where peptide bond is being formed, which is at the larger subunit where the enzyme peptide transferase is present. And after peptide bond formation, you can see that the amino acids are transferred to the tRNA, which is at the A site. So in our case, what happens is we are with the starting point of the translation. So what happens is after the first peptide bond formation at the P site, you will have a tRNA which does not have any amino acid, and the tRNA which is at the A site will have 2 amino acid, because the amino acid is now transferred to the tRNA which is at the A site. So this transfer is also catalyzed by the same enzyme which is called as the peptide transferase. So this is the main event that occurs during elongation process, which is called as peptide bond formation. So after peptide bond formation, you can see that the amino acid are transferred to the tRNA which is at the A site. Now the tRNA which is at the P site is devoid of any amino acid. So moving on to the next step, which is the process called as translocation. So translocation means after the peptide bonds are formed, the ribosome will move 3 nucleotides towards the 3' direction. As we said earlier, it is moving on in the 5' to 3' direction. So the ribosome moves 3 nucleotides towards the 3' direction. When this ribosome moves to the 3' direction, the particular process which is called as translocation occurs. When translocation occurs, as seen in these particular videos, when translocation occurs, you can see that there is a change in the sites. So this one also clearly, these 2 videos, it is clearly depicting the process. So see this particular video, you can see after every peptide bond is being formed, there is a translocation where the mRNA is being moving 3 nucleotides each. As a result, new 3 nucleotides are reaching the A site. So now what happens is new 3 nucleotides reaches the A site, which is devoid of any tRNA. Now our tRNA, which was previously at the A site with a dipeptide, will be shifted to the P site. That is being shown here. You can see the tRNA with the amino acid is changing its position from the A site to the P site. So every time it occurs, you can see the tRNA with the amino acid will be shifting to the center point, which is called as the A site. And now the particular tRNA which was present in the A site, P site, which was devoid of any amino acid, because it gave the amino acid to the other tRNA. What happens is that as you can see in this particular diagram, there is a small kink or a small fold. That means there is a cavity. That means the mRNA is now looping outside. As a result, whenever the tRNA without an amino acid reaches the E site, it will lose its complementarity, because there is the complementarity, the codon-anticodon pairing is the one which is holding the mRNA and the tRNA. And whenever the tRNA, because the entire mRNA is moving, and when it moves, this particular codon, its complementary codon also moves with it. But when it reaches the E site, because there is a kink, because the mRNA is now moving downwards, the pairing will be lost. Whenever the pairing is lost, the tRNA will move outside. That means a deacylated. Deacylation means it is a particular tRNA without an amino acid. The deacylated tRNA will shift its position during translocation from the A site, so from the P site to the E site. When it reaches the E site, because of a kink in the ribosome, the mRNA, the complementarity or the codon-anticodon pairing will be lost, and this loss in codon-anticodon pairing will cause the tRNA to move out of the ribosome. Now the process will continue. The new, as you see in these particular videos, a new tRNA is coming at the A site. Now the particular tRNA which is at the P site with 2 amino acid, shift it the amino acid to the one in the A site. Now the A site tRNA will have 3. Then translocation occurs. The tRNA with 3 amino acid will come to the P site. The other amino acid, which is tRNA, which is deacylated, is moving out of the ribosome because of the kink. So this particular process will continue till our ribosome reaches a position, and that position is nothing but we are going to see the next step, which is the last step, which is called as termination. So termination occurs whenever so translocation is happening, peptide bond formation is occurring, etc. And whenever the A site reaches a particular sequence or a particular codon, which is called as a stop codon. It can be any of the 3 stop codon. When it reaches there, there is no tRNA which is complementary to the, which is having an anticodon which is complementary to the to our stop codons. And there are some factors here which helps in termination. They are called as releasing factors. There are 3 releasing factors: RF1, RF2 and RF3. And the 3 releasing factors, these releasing factor in this particular termination, they take up the role of the tRNA. They take up the role with tRNA means they cannot form a peptide bond, etc. They are going to terminate this particular mechanism, or they are going to terminate this particular process of translation. So what these releasing factors are going to do is can be seen in the slide, because the releasing factor, the first releasing factor, which is RF1, this particular RF1, it can recognize 2 stop codons. RF2 can recognize another 2 stop codons. RF3 is non-specific. It can recognize any stop codon. Recognize in the sense, when a stop codon comes at the A site, that means now the open reading frame is at the end, or the mRNA, the translation process is to be completed or terminated. So any of the 3 releasing factors, depending upon their whether it is RF1, RF2 or RF3, it joins or it will move into the particular stop codon region or into the A site, where the releasing factors binds to the A site. Now this peptide transferase, which is present in the larger subunit, it will search for an amino acid to form a peptide bond. And whenever an amino acid is not present, what peptide transferase does is, that means whenever in the A site a releasing factor comes, that means there is no amino acid in that. What peptide transferase does it will cleave off the polypeptide from the tRNA which is present in the P site. So now the P site is having a long polypeptide chain, and usually what happens is, if there is another codon, this long one should be transferred to the A site. Now it cannot be transferred, because instead of a tRNA, you are now having a releasing factor over there. As a result, there is a releasing factor about peptide transferase. The same enzyme which is causing the peptide bond formation causes the cleavage of the polypeptide from the tRNA. As a result, the entire polypeptide will be released as a protein towards the cytoplasm. And whenever this polypeptide chain is broken, the tRNA and releasing factor, etc., they move out of the ribosome. The 2 subunits of the ribosome, they separate from the mRNA, and this marks the end of what is called as a process which is called as translation.